Hello. Hello, everyone. Good morning, good afternoon from wherever you are joining us from. Thank you for taking out time to join the webinar today. It is just a minute before the scheduled time. So I just came on here to acknowledge all of you and say thank you for joining us. And we'll start at, let's just give others 2 minutes to join. Right now we have 33 attendees on the call. So we'll start at exactly 2 minutes after the scheduled time. Thank you so much. Once again, thank you, everyone. It's at exactly 2 minutes after the scheduled time. We already have over 50 people on the call. So we'll just go right ahead because we have quite an interesting presentation lined up today. So I'm Oluton Sanusi Oladuni, and I am your moderator for this critical discussion. As lead of the African Vaccine Academy at Bloom Public Health, I see firsthand that robust regulatory systems, especially bioequivalence, are foundational to Africa's health security and access to quality medicines. These standards build public trust and are essential for our continent's pharmaceutical future. So welcome again to this crucial discussion on removing roadblocks to quality medicines in Africa, understanding mandatory BABE and expert solutions. Through our bioequivalence training initiative, the collective mission is to empower stakeholders and facilitate the implementation of robust BABE standards that align with global best practices tailored for the African context. A few of our service offerings include comprehensive GCP, GLP training and workshops, expert review of BE study protocols and final reports, and also support for regulatory authorities in assessing BE study reports and reducing dossier backlogs. The team are experts who bring a wealth of experience across the regulatory, scientific, clinical, and international domains of bioequivalence. Allow me to briefly introduce a few of them. We have Dr. Patrick Lukolai, is an authority in pharmaceutical sciences and clinical pharmacology, providing deep scientific and technical guidance essential for robust BE principles and studies. We also have Professor Chimeze Ayankora. He is the CEO of Bloom Public Health and a strategic leader in capacity building and resource mobilization. Also, Dr. Lois Kikwai is an expert in African regulatory affairs and market access. We also have Dr. Marina Fetek, who brings a crucial international perspective on global regulatory standards and best practices. Our core presentation today will be delivered by Dr. Lois Kikwai and Dr. Marina Fatek, who will delve into the strategic importance of mandatory BABEs and the pathways to overcoming common implementation strategies. Following that presentation, we will open the floor for questions and answer session to address your specific challenges and roadblocks. Kindly drop your comments, your questions in the question and chat boxes below so my team can pull them up at the end of the presentation. Having framed the landscape and the imperative for quality, let us now move to the core concept that will help us understand how to remove these roadblocks. I hand the floor over to Dr. Lois Kikwai and Dr. Marina Fatek for that presentation. Over to you, Dr. Lois and Dr. Fatek.
Thank you, Olutun, for that introduction. Marina? Has Marina been able to join?
Yes, she has.
Okay.
Okay, so she asked me to unmute her mic. I'll do that immediately. Okay, you have access now, Dr. Marina. Please confirm if you
Hi,
could.
yes. Thank you so much. Welcome everyone to this webinar, and thank you for having me here today. It's a pleasure.
So Marina and I have a couple of slides to really introduce the topic on understanding the mandatory BE requirements. As you all know, the session is scheduled for about an hour, so the presentation will not even cover all of the requirements that are needed to be able to execute BABE studies. However, this is a starting point to allow us to see what is required, why is it important as a key discussion point, and also why has it become so such a hot topic right now on the continent in terms of meeting the requirements for BE. What are the regulators asking for? What is industry supposed to be doing? And then what are those key stakeholders in the whole spectrum where BE is required? What is everyone supposed to do it and how to accomplish it to meet all the requirements? So as we get started, the slide is removing roadblocks, or the topic of our presentation is removing roadblocks to quality medicines in Africa, understanding mandatory BABE and expert solutions. So next slide, please. So this slide just serves as an introduction to our moderator and the speakers, myself and Marina. I think Tone has done justice to the introduction, so I will not add anything on about myself, and we'll go straight to the presentations. Right, Marina?
Yes, agree.
Okay. So the topics that will be discussed today include the impact of generics on healthcare, short history on bioequivalence, and this is relevant to what has happened in other places. And then we can align it to what is happening on the continent right now and see that we are on a path that has already been traveled before. So there are many options in terms of what we can do as industry regulators and those stakeholders interested in this topic. We'll review some regulatory landscapes for generic drugs. What are the international guidelines? What do we have on the continent and the current situation? And then now we'll go a little bit technical and introduce bioequivalence and what are the key considerations for bioequivalence and some challenges. And then we'll open up the session for some questions and answers. Okay, next slide, please. So, what is the impact of generic drugs on healthcare? As we know, generic medicines are integral in modern healthcare. We see across the globe, whether it's on the continent, whether it's in the US, whether it's in Europe, that over 70% of medicines are generic drugs, and they take a market share value of about 20%. And this is because generic drugs come at a lower cost than brand name drugs, but there is a reason for this, which we'll discuss a little bit later. Generic drugs are available in all forms. They can come from the basic, what we know in the day-to-day solid or dosage forms, liquids. However, they can even be more complex and tackle more complex diseases such as antimicrobials, immunological drugs, and also their formulation can also be complex in their nature. And so their development may require a bit more intense scientific proof and rigor during regulatory evaluation. The value of generics, as we said, they come at a lower price. They have so they're economical. As we see, over 70%. So we have patient access, market access to these generic drugs. They're available easily on the supply chain, and some of the generic drugs also include innovations that make them easily available and easy to use by the patients. Generic drug applications are termed abbreviated or shortened because they generally do not require to include longer preclinical and clinical studies. And also this main requirement also affects the economic aspect of it, where because preclinical and clinical studies have not been done, they are cheaper economically. However, that does not mean we ignore the safety and the efficacy of these generic drug products. They basically rely on preclinical and clinical data that has already been submitted for the brand. So as we know, generic is a comparison between a generic drug and a brand drug. They have the same quality and they should have the same expected safety and efficacy by relying on this preclinical and clinical data. So basically, generic applicants must scientifically demonstrate that their product is bioequivalent. What does bioequivalent mean? It means same safety and efficacy. And how do you show bioequivalence or something that is bioequivalent? By conducting bioequivalent studies. And these are equivalency studies that are conducted in the body, in humans, and hence the bio aspect of it. Next slide, please. I don't know how clear this slide is. I hope you can all see it, but it just basically offers us a pathway to show the history, some principles and background of bioequivalence requirements. We see that, and this is relying on the history that has been seen in US, where in the early 70s, there were a lot of reports of intoxication, variability, and poor effects from various drug classes, including NTIs. The regulatory authority then in the US developed the Hatch-Waxman tongue twister of a word, drug act, where the drug pricing, competition, and patient term resolution was evaluated. And in this act, we see that establishment of ANDA pathways for generic drug was also included. That means it simplified the approval based on pharmaceutical quality, and there was no in vivo BE studies that were required. However, in the 80s, we started to see concerns about drug substitution and discussions about generic drugs and are they as good as brand name drugs. And so this triggered a bioavailability bioequivalence committee in the late 80s, 1986, where we found that it was necessary to define what is bioequivalence, but also define the bioequivalence statistic procedure to be able to evaluate bioequivalence study. And so there was a task force that was formed, and we see that this task force was created to review how generic drugs are approved, whether it's taking into consideration the quality aspect or the bioequivalent aspects. And here we also see there's a lot of statistical evaluation was highlighted as it relates to the issue of bioequivalence, because you are basically comparing two things, and you have to ensure that you have the right statistical evaluation and how the studies are designed. So from then we started to see various iteration of FDA guidelines on statistic for bioequivalence, guidelines that relate to evaluation of bioequivalence studies. And the more we learned about bioequivalence, the more advanced the guidelines became. And we are at a point 2024, 2025, where we actually have harmonized BE requirements and guidelines for industry on how to conduct bioequivalent studies for solid oral dosage forms. However, if you look globally, and Marina will discuss, you'll see different authorities already have guidelines for this topic. Next slide, please. So the key stakeholders in bioequivalence and conducting and generating bioequivalence study and consuming products that are generic drugs that have been considered with good quality, safe, and effective there are various stakeholders: pharmaceutical industry, who make the products and do all of the testing; contract research organization that assist this industry by conducting some of the clinical and bioequivalent studies; medicines regulatory authorities, who evaluate the data that's generated by these two organizations, both the quality side and the clinical side; and then we have the public, the public health, who consume these medicines, generic medicines that are produced. Next slide. So everyone has an interest in what's going on. I'll give my colleague Marina to talk about the different regulatory landscape and the different guidelines that are available. Thank you.
Okay, thank you, Lois. So now we are going to take a look into the global development of the regulation and guidance for regarding bioequivalence. So for us, we have chosen these three: US FDA, European EMA, and WHO guidance, because they are the three largest and most impactful jurisdictions, and they're also jurisdictions that have enormous experience with generating the data for bioequivalent studies, conducting bioequivalent studies, and also assessing bioequivalent studies as a part of a drug approval. So there are significant similarities in these three major guidances, but there are also some differences. So US FDA has a guidance for industry regarding bioavailability and bioequivalence studies for orally administered drug products. There are also food effect bioavailability studies, or description or guidance how to do them, what is expected, and which data should be generated to enable meaningful assessment and successful approval. EMA has a guideline on the investigation of bioequivalence. It also gives instructions similar to US FDA, how to conduct bioequivalent study, which type of bioequivalent study is appropriate for specific dosage forms. Then there are also references to other studies, because we need to see these guidances as there's basis for other relevant guidelines which are derived from bioequivalence guidelines. So in US and in Europe as well, the industry is mainly guided by these. However, CROs or sponsors or generic companies, they need to always take into account, they need to be aware of the specificities of their own product. They need to be aware of the claims that they are making for their own product, and the development of these products should be adjusted to these claims. So these are basic, very general recommendations. And then there is, of course, WHO technical report series, which is also providing instructions how to conduct and generate data for bioequivalence, and also give some criteria what is actually considered bioequivalent. So next slide, please. And then we have ICH, which is a very huge and very influential organization. This is an international conference on harmonization. And their aim, it was founded by US FDA, European Commission, and Japanese regulatory authority, and then other authorities, and also the industry, of course. There are industry stakeholders in the ICH as well. And the aim of the ICH is to provide one single guidance that should be acceptable to all jurisdictions which are part of the ICH. So as I said on the previous slide, there are differences between EMA, US FDA, and WHO, and the aim of the ICH is to unify or to erase these differences and offer one single guideline which can be applied to any jurisdiction. And this ICH is particularly important for the companies or sponsors which are developing drugs, for example, for European market, for US market, for African markets, for Asian markets. And to erase the differences, then they can take ICH into account and adjust their development according to these guidelines. So for us, for bioequivalence, important guidelines, the ICH are the ICH-M9, which are BCS-based biowaivers, ICH-M10, particularly important. This is bioanalytical method validation and study sample analysis. And this ICH-M10 is a very technical guideline, and it actually provides instructions how the bioanalytical method should be validated, what is expected, and how the sample analysis should be performed, and how the data should be presented. And then, of course, we need to take into account guidelines for good clinical practice. This is ICH E6, and of course E9, statistical principles. And then we have M13A, M13B. These are guidelines, and there was a lot of talk about it last year because there was a lot of work was put behind updating these guidelines. And there were many meetings of the ICH group, and they finally, I think in October 2024, they came up with a new version of the ICH guideline, which provides a recommendation for oral immediate release solid oral dosage forms, provides recommendations for biowaiver, and compared to what we have had before, this guideline basically brings closer together European and US FDA guidelines. So next slide, please. So, and now we are going back to the African continent. I will go briefly through these. There are in Africa, regulatory authorities, they have their own publications. We have here Egyptian, Ethiopian. Then if we go to the next slide, then there is from Rwanda FDA. So what should be the aim here? I can talk about experience in Europe. Because Europe is very similar in its structure to African continent. We have national competent authorities and then we have European Medicines Agency. And in order to provide good quality generics to the general public, the aim of the European Union was also to provide or to enable transparent and consistent assessment across the Union, which means that we needed to have unified guidelines. So EMA, actually, or EMA working committees, they are the ones who are issuing guidelines, which is the, for example, bioequivalence guideline or bioanalytical guideline that we use. And all the national competent authorities, they are driven by that guideline in their assessment. All sponsors that are trying to submit application in Europe, they are conducting their research and their drug development according to these guidelines. This has proven very successful because the impact of this unification on the generic market and ultimately patient access to cheaper and good quality medicines, that is generic medicines, has been tremendous. So it took a long time, of course, after the formation of the European Union, but I think we are at a very good place right now, and this, from the regulatory perspective, there is a lot of trust from the industry into the transparent and good quality assessment from the European regulators. Of course, the drug marketing authorization is only one step of the regulations, and then there are national specificities. Of course, there is a specific needs of certain patient populations and specific needs of certain countries. For example, when it comes to medicines shortages, and they are handled on a national level. Pricing and reimbursement is also handled on a national level. But when it comes to drug marketing authorization, to make our European market more attractive, there was a huge effort to unify and make the assessment transparent, which will drive the product development from the sponsors in a more streamlined way. So I think this can be a very good example also for African continent, how to improve the innovation on the continent, how to improve the assessment on the continent, and how to improve the regulatory environment on the continent. So next slide, please. Okay, so I will hand over to Lois because she is definitely a better person to talk about the situation in Africa. So Lois, please go ahead.
Thank you, Marina, and thank you for the segue in bringing up the EMA situation in terms of different countries and different NRAs organizing and working together in the central EMA pathway, and as it pertains to general regulation and marketing authorization of generic products. So as you can see from Marina's example and also our earlier discussion, where we were talking about the history of BE, we see that we had to start somewhere. There was something that triggered the need to make stringent regulations around generic drugs and around what is required to ensure a generic drug is safe, good quality, and efficacious. On the continent right now, I think the major driver of this situation is the efforts of regulatory authorities being evaluated under the WHO Global Benchmarking Tool and being given different levels of maturity level 1, 2, 3, or 4. And in these efforts, one of the key requirements in the GBT is that the authority must make sure that the products being sold on the markets have good qualities, have safety, and efficacious. And specifically as it relates to bioequivalence, we see that in various places in the GBT, it quotes that in both instances where you have legislation, the first requirement, and also guidelines, and ensuring that you have guidelines for the products that are for marketing authorization. In both instances, it says that the products multi-source product should demonstrate bioequivalence or bioavailability when it is deemed necessary. So as you can see, in terms of enforcement, maybe this was not fully enforced by various regulatory authorities on the continent. However, now it's become a requirement to ensure that the enforcement is implemented by the authority so that they can function as a mature level 2, level 3, level 4 maturity level authority. And so this is basically where it's coming from. And it's not coming because it's just an additional requirement that needs to be done. It is critical to be able to determine whether the products are, especially in terms of safety, and if it's performing well in a bioequivalent manner in the body. Next slide, please. So with the efforts of different regulatory authorities to achieve maturity level 3, we have some that have already achieved maturity level for medicines and vaccines producing, and for vaccine producing and non-producing countries. So we have Ghana, Nigeria, Tanzania, Zimbabwe, Senegal, Rwanda, Egypt, and South Africa. These are all authorities that have been evaluated according to GBT and have received a level of maturity as it relates to the WHO GBT. From these countries, we see that we are also getting published information and circulars and memos from the authority that BE is now going to be enforced if it wasn't being enforced, or more data is going to be needed by the different authorities. So the next couple of slides, if you go to the next slide, please, shows recent communication from the regulatory authorities as it pertains to bioequivalent. Here are some communications that you can find on the NAFDAC website as it relates to the guidelines on investigating bioequivalence, the regulations for bioequivalence, the notice to industry that bioequivalence is required and how they should proceed, press briefing related to bioequivalence, and this is from NAFDAC. We know NAFDAC is a maturity level 3 authority. Next slide, please. We also see communications from the Kenya PPB, Poisons Control Board. And here we see there are various validation meeting. Even I think more recently, Kenya has been designated as a center of excellence as it relates to bioequivalence. So in terms of striving to accomplish this requirement according to GBT and basically according to global regulations, there are efforts from different regulatory authorities. Next slide, please. We also see there on the Tanzania website and on the South African SAPRA website, there are links to guidelines and documentations and communications to industry as it relates to bioequivalence. Now, this is just a small sample of what is going on the continent as it pertains to bioequivalence, but then there's also a lot of questions in terms of, as industry, as local manufacturers. BE was not required before. What do we do now? Where do we start? And our message today, and as titled in our presentation, it's removing those roadblocks. Some of those roadblocks is we don't know where the guidelines are, we don't know the templates, we don't know how the CROs will work. So we're trying to bring to light that a lot of this information, guidelines, regulations are either available on your country regulatory authority website. However, there are other global organizations where you can find the same instructions as it relates to bioequivalence. As you can see, the history of bioequivalence did not start yesterday or 2 years ago, 3 years ago, but it has come from the 70s. And so there's a lot of information out there. Next slide, please. Actually, this would be a good point to end in terms of regulations and requirements and switch our talk a little bit to really what is bioequivalence, what is required to say that my product is bioequivalent and I have enough data, I've generated enough information that's satisfactory to the regulatory authority to issue a marketing authorization for my generic drug product because I've met all of the requirements. So I'll switch back to Marina, my colleague, to discuss some of the key requirements for bioequivalence. And also she will finish off with some challenges that face practitioners as they perform bioequivalence studies.
Okay, thank you. Thank you. Okay, so in order to talk about bioequivalence, we first need to understand what is actually bioequivalence. And this is actually the definition that you see here on this slide. This is the definition that was taken from the European legislation, and it gives a definition of a bioequivalent product or generic product. So if the two products, they have the same active substance, they are considered bioequivalent if they're pharmaceutically equivalent, or they have comparable in vivo performance. So what does that mean? That means that basically EMA or European legislation is opening a pathway to product marketing authorization in two ways. One of them is pharmaceutical equivalence, another one is in vivo equivalence. This is, of course, very important because there are products for which there is no need to do bioequivalence studies. For example, we are talking about eye drops solutions. We are talking about, for example, some topical products where it is not possible to demonstrate bioequivalence. We can talk about the BCS biowaivers. That's a special topic. We are not going to cover it today in detail. So all of this needs to be anchored in the legislation, because actually drug regulation, it is sort of an interface between science and regulation. It is an interface between main stakeholders, between the industry, which is always driving the innovation, between CROs or contract manufacturing organization. They're the people that are actually, they have hands-on experience in conducting bioequivalence trials. And then there are regulators which are saying yes or no to which products should be allowed on the market. So all of this needs to be somehow defined, and there should be some sort of accountability for each of these parties. And the legislation and the definition of bioequivalence is what we are talking about today. So you can see here that, for example, in European legislation, there is a special article. It's called Article 10 of the directive. And it's important to say, when we were talking about harmonizing the processes across countries and continents, the directive 2001 by European Commission is one piece of legislation which is valid in all EU countries. So every country which belongs to EU, of course, has a national regulatory authority. They are driven by the articles and laws which are in this directive. This directive, it's translated into national languages, and also all of the most of the, no, actually all of the aspects that are in that legislation are part of national legislation. So the purpose of establishing bioequivalence is to demonstrate equivalence in the biopharmaceutics quality between the generic medicinal product and the reference medicinal product, in order to allow bridging of preclinical tests and of clinical trials associated with the reference medicinal product. So what does that mean? This means that a company comes, they have a new active substance, they do a lot of preclinical tests, they characterize the pharmacokinetic behavior of the active substance, of the dosage form, they perform efficacy and safety clinical trials, and the drug is approved with some data and market exclusivity. Once the data and market exclusivity expire, then generics are allowed to enter the market. But in order to prove that this generic will act in the same way as the reference drug, bioequivalence needs to be performed. That's the abbreviation in the whole process of the drug development. And once we establish that bioequivalence, that these products are bioequivalent, we can say that the efficacy and safety data of the reference of the originator product are bridged to the generic product. We have the assurance that efficacy and safety of the generic will be exactly the same as efficacy and safety of the originator. And again, all of these principles, they're outlined in EMA guideline on the investigation of bioequivalence or US FDA guidance for industry on bioequivalence. So next slide, please. So now we're coming to the practical aspects of designing the bioequivalence study. And on this slide, you can see actually all of these things that are written on this slide, they are taken from the guideline. So the guideline gives you a choice, which type of study, crossover, replicate, parallel. How many subjects do you need? Healthy volunteers? Do you need patients? Is the study going to be fasting or is it going to be fed? With what will be the reference product? That's why we were talking about full data set of clinical efficacy, clinical safety, toxicity, pharmacokinetic characterization, and so on. So in European legislation, for example, reference product needs to be exactly what I was saying. It has to have a full data package and it should not be under market and data exclusivity. Then PK metrics. There are some standard PK pharmacokinetic parameters that every bioequivalence study should have. And then there are some specific PK parameters, and they will depend, again, on the product that is being developed and on the claims of the company, what this product should do. So according to this, companies will choose to add some pharmacokinetic parameters like partial AUCs, Cmax at steady state, or T-lag, which is the lag time until the onset of effect, for example, and so on and so forth. Then bioequivalence limits. It's not one bioequivalence limit. Usually we take bioequivalence limits as confidence interval, 90% confidence interval to be between 80 and 125, but that is not always the case. There are some products which have more narrow confidence intervals. There are some products which have wider confidence interval. And this is something when the drug product is being developed, this is something that should be kept in mind because it needs to be pre-specified how to deal with statistics of bioequivalence. Then, of course, safety monitoring or patient safety or subject safety and data integrity, they're a very important part of the bioequivalence trials, and they are part of the GCP, good clinical practice, and they're subject to national authority inspections. And these are one of the key things when, for example, new bioanalytical or new clinical site is being, developed. These are the first thing that should be taken into account. How to set up systems which will enable data integrity, which will enable tracking, documentation, quality systems, and patient safety. So. Some of the other things to consider here when designing and conducting a bioequivalence study. First thing, which is actually the subject of the bioequivalence study, is the formulation itself. because when formulation is being developed in the laboratory, when it's being tested, there are usually several prototypes of the formulation, and the most optimal one is then, carried forward into the clinical or PK bioequivalence program. So formulators in the labs, they need to think about the composition, they need to think about excipients, they need to think about patents that are out there, not to breach any patents. If someone is trying to, develop a different dosage form, then how to, conduct bioequivalence to prove or to characterize this different dosage form, what can you expect? So these are some of the things that sponsors and CROs should think about in advance when designing bioequivalent studies, because all of these formulation characteristics, they will be somehow seen in the bioequivalent study, and they will be reflected in the data that are generated. Also, what is important is the choice of a comparator product or reference product. From a formal point of view, there is a slight difference between a comparator and reference in the European legislation, but in the context of a bioequivalent study, we can say that it's the same thing. And basically, this is a product that has a full data package, as we were talking about it before. And in EU, for example, there is a specificity, and I think it's also valid for US, that the reference product should be sourced from EU market. Why? Because it can happen that, for example, in global development, there is a manufacturing site, one in Africa, one in Europe, one in India, and the way the reference product is manufactured and the way excipients are used, the grade of excipients, the purity of excipients, it can affect the reference product. And it can happen that the reference product from the US market and European market is not bioequivalent to each other. So that's why Europe said, okay, fine, but we want to see reference product from our own continent. It doesn't matter from which country, but it should be from from EU market.
Marina, I can also,
yeah.
can I jump in a little bit? In terms of comparator, I think also the same considerations are now happening within some jurisdictions on the continent where, yes, they want to see the international comparator, but if that product is, the brand product is on the market within their jurisdiction, there's also some requirement of, depending on the type of studies required, from dissolution to even more extensive studies, where you have to show comparability with the product on the market. One of the big things, especially that we were saying with this, generic drug products, is they're supposed to be interchangeable. So yes. I'm not interchanging with something that's in a different market jurisdiction. It would be interchanging with something that's within my jurisdiction. So this is one of the things that pharmaceutical companies, especially local manufacturers, have to take into consideration. What is your product being interchanged with from a generic to a brand? And selecting and choosing a comparator product is a big topic. I think there's also some WHO guidelines or guidance on that. So I just wanted to jump in. Also, same principles apply also on the continent.
Okay. Yeah. So I mean, even in Europe, the comparator or reference product, it can be a problem sometimes, and I have seen it in as a consultant that, for example, when companies go to a scientific advice, when they start communicating with the regulatory authority regarding their development, very often the one question is to the authority is this reference product acceptable for bioequivalence study? And I do very often advise companies to put in this question if the reference product is not straightforward, or if they are not sure that this can be used, because actually the health authority is the only body that can say with certainty yes, this is good reference, or no, this is not good reference, because they have all the data that we do not see all the time. So another important thing is study population. We have to take into account, first of all, do we need to enroll patients, or can we enroll healthy subjects? When can we not enroll healthy subjects? There are some drugs, for example, antipsychotic medicines that are not tolerable for safety reasons. They cannot be given to healthy subjects. There are some anti-cancer medicines that cannot be given to healthy subjects. So patients need to be recruited. There are also, we need to think about genetic differences. What I'm seeing sometimes, for example, Asian developments from, say, Korea or from Taiwan, they perform bioequivalent studies on Asian patients or Asian subjects. But when they bring this product to Europe, how can we be sure that the pharmacokinetic will be the same in Caucasian population? How can we be sure that when the study is performed in Caucasians, that it will be applicable, that it will behave in the same way in African population, because we do not have the same genetic predispositions. So this is something which is very important to take into account. It's important for global developments. It's important for CROs to know what are the aims of their sponsors. For example, if an African company wants to market in Europe, they will need to take into account that their data may not be acceptable because they're not able to explain that there are no genetic differences and that African data will be applicable to Caucasian patients. So study design itself depends on the properties of the formulation. We have talked about this. Depends on the variability of the active substance, and this is also something to think about, because we can talk about the power of the study, which means how probable it is that the bioequivalence is true, that it's not a fake result. So from this perspective, study design is very important. Also, number of subjects, then appropriate bioanalytical method. It's a big topic on its own. It needs to be validated, and it needs to conform to certain criteria, and also it needs to be very well documented. Then also sampling time points. Some of the products, they're straightforward, but for example, inhalational products, they're technically very difficult to take samples from, because if Cmax, maximum concentration, is reached in 5 minutes, then the first sampling point should start somewhere, say, in minute 1 or minute 2. After inhalation, which is very difficult to achieve. And I mean, not every CRO is able to do, for example, such studies. So from the sponsor's perspective, the bottom line is depending on the product that is being developed, depending on the claims, the choice of a CRO will be also very important. What they are able to do, how they are able to recruit patients, what are their experience with some, say, specific dosage form. And of course, the ultimately is ensuring data integrity and GXP compliance. That's the job of every CRO. And as I said, these are subject to inspections by regulatory authorities. Okay, so next slide, please. Okay, so some of the challenges of the data analysis. We will go through these quickly, but I just wanted to, give an example of something that we see very often as consultants, that something that we help our clients with, something that regulators very often see on their desks, and something that they will most definitely ask. So one example is that, for example, 90% confidence interval is slightly outside the pre-specified margins, which means that the bioequivalence failed. But how to interpret this? There are several ways to go: to repeat the bioequivalence study, to do something differently, to say this is not relevant, or to say this is a no-go, this product is no good, we are not going to approve it. So the decision to repeat the bioequivalence study depends on the reasons for failure, depends on the root cause analysis should be performed in these cases. And then we need to ask ourselves, if we do repeat the study, are we increasing the probability that bioequivalence will be fine, or that we will achieve bioequivalence, or is there something else happening that is preventing me from showing bioequivalence? Is it something in the formulation? Did I recruit enough subjects? So all of these things, they are common challenges that we see in the bioequivalence studies. For example, for inhalational products, did I train my subjects well how to inhale? Because if they didn't, they will not achieve the concentrations that I need. So, and also ultimately, what is the clinical relevance of the bioequivalence study results? Sometimes, even if bioequivalence is not achieved in some parameter, I gave an example of Tmax for tadalafil, we can offer an explanation why it doesn't matter that it's not bioequivalent, that it's not absolutely bioequivalent to the reference. Why? Because, for example, for tadalafil, we cannot find the PK/PD correlation, which means no matter what the behavior is from the pharmacokinetic perspective, efficacy and the pharmacodynamic effect will be different. So, but of course, tadalafil is one example. There are examples where it is not possible to justify anything at all, and it's just necessary to either repeat the study or from the point of view of the regulators to reject such an application. Then one other important thing is it's a very big challenge for the regulators. It's a big challenge for CROs and also for sponsors. Ultimately, is the GCP non-compliance and GCP fraud detection of the CROs. In Europe, every couple of years, inspectors, they go out, they inspect a whole bunch of CROs, and there is always one CRO which is GCP non-compliant. And the consequences of this non-compliance are tremendous, because if CRO is not compliant, data integrity is compromised and we cannot trust bioequivalence. What does this mean? That all products that were tested in that one particular CRO are withdrawn from the market, and sponsors, they need to present data from another study that they have performed on this product. This process is very tedious for regulators. It's very tedious for the sponsors, with very uncertain results. So that's why nobody likes GCP non-compliance. Nevertheless, it happens all the time, and this is something that we need to be very, very much aware of, from the point of view of regulator and from the point of view of CROs and sponsors. So next slide, please. Okay, so from the regulators' perspective, I mean, Lois and I both worked as regulators before we became consultants. So I think we have a very good perception of what it means to regulate medicines, and it always means a balance between public health and public interest. So what should the regulator do? They should ensure that medicines that are brought onto the local market are of good quality, of acceptable quality for, a large patient population to use them. But there is also public interest, because public interest is to bring good quality medicines for acceptable prices, to lessen the burden on the healthcare system, and generics are a perfect tool to do that. But safeguarding public health should not be an excuse to pose challenging regulatory hurdles, and then as a consequence of that is actually to neglect public interest. So this is a very, very, very sensitive interface, actually, this public health versus public interest, and it always needs very careful balancing. And this is the regulators, primarily regulators' job to do, which means communicating with the industry stakeholders, and maintaining transparent and streamlined regulatory processes to enable transparency and consistency and predictability in the assessment. The industry job is to do innovation and challenge the regulators with new and innovative ideas. So which means that there is also balance between science and valid guidelines. We all know that guidelines that are updated more slowly than the science. Then science is very quick, and that's why I said it's a job of the industry to challenge the regulators with this new scientific approaches, new scientific ideas. And it's a job of a regulator to put it into the right balance with what we know and where the risks are. So another challenge is first-time generics. For example, when the market exclusivity expires and the first generic comes to for assessment, regulators, they don't have any experience assessing such generics. So there is always a question, how much data do we need? What kind of data do we need? How good quality data do we need? How much experience can we take from the originator assessment? So I know that in Europe, for example, first generics, their assessment is very rigorous and it's more conservative than it is for 150th generic. So after some time, when the regulators, when they collect enough data, they can say, yes, we are sure. Now we can let go of the strict regulatory rules. So, and then there is a requirement to repeat the BE study. That is also a big challenge. Sometimes my perception from the European regulation that this question comes too easily by the regulators, but I said many times before, and I'm saying it now, we need to also keep in mind that there is blood behind every bioequivalence study which is taken from subjects, which is taken from patients. So repeating bioequivalence trial, it's not an easy decision to do, and it should be really considered very carefully and taking into account the ethical aspects of the study, taking into account, do we really need new data, or is it absolutely necessary to provide the data? So to conclude, we are, I think, a little bit behind the schedule. So bioequivalent studies are the foundation for ensuring access to medicine with significant positive impact on public health, which means providing more medicines to more people, providing good quality medicines for lower prices. In many jurisdictions, BE concept serves as a surrogate for efficacy and safety of generics. It is anchored in the legislation. It is harmonized across European countries. In US, it is only one US FDA, so it is harmonized there. And the requirement to do bioequivalent studies will drive the industry, it will drive the innovation, and vice versa. Conducting of the BE studies will define the regulatory environment. So that's why we are emphasizing a lot that streamlining and making the regulation transparent and predictable will this means that the market will become more attractive to the industry and it will drive the generic development. And then challenges of conducting, analyzing, and assessing a bioequivalent study are unique for each product. There is no repetition of the same situation. Each product is different and it should be approached individually, taking into account previous experience with similar products, taking into account valid guidelines, and taking into account current scientific approaches. So this is all from us. Thank you very much. And I think we can take a couple of questions quickly.
Thank you so much. Thank you so much, Dr. Kikwai and Dr. Fetik, for that valuable presentation, highlighting key challenges in the African bioequivalence study. So we'll now open the floor for questions. So this Q&A session is the audience direct channel to our experts here on the call. Please submit your questions using the Q&A feature or drop them in the chat box. So a few questions came in earlier, and I would ask our experts to please shed more light on it. So facing diverse national requirements and the need to align with international standards, as we've seen, we have the WHO, the EMA, the ICH, while leveraging on AUDA, NEPAD's harmonization efforts. What are the most effective strategies for streamlining the BABE submissions in Africa for pharmaceutical companies and regulators?
So, Joan, the last part of the question is, what are the strategies?
Yes.
For streamlining?
What are the strategies? Yes.
I think one of the things that's come up from this presentation is the fact that we do already have guidelines that have been developed, have been used for many years. I do also want to say that on the continent, we have a lot of harmonization efforts to be able to streamline some of the guidelines and requirements, especially on the regional block area with AMRH. And also now as we move towards AMA, there are already harmonization efforts that are taking place. For these harmonization efforts, I think one of the key things is rely on regulatory guidelines that are already existing. Bioequivalence is not new. And so starting from the ICH guidelines that talk about solid oral dosage forms. However, there are guidelines for other types of generic products, depending on the product type, the formulation type, the complexity of those generic products. So I think we have to keep abreast on what has already been published. And then also another strategy is what is relevant for Africa? What do we need as Africans to make this bioequivalence relevant for our continent, the type of products that we receive, and the type of studies that we want to conduct for our continent? I think Marina had mentioned, genetic differences and things like that. So those are some of the things that need to be taken into consideration. These will be studies for products that will be marketed on the continent, and they should be relevant and strategically designed for our population using guidelines that are already there on the global space. Thank you. Marina?
And if I also may add one thing, I think what is also important, that even for this harmonization, is an intensive dialogue between the industry, between the CRO, and between the regulators, because sometimes it happens that one CRO actually has more insight into specific requirements of each national authority than national authorities among each other. So, and we know also from, for example, from bioanalytical inspections from that CROs undergo by US FDA and European authorities, that they can be different and they can go against each other. So I think from this perspective, the conversation should be intensive, and it should be ongoing. It should be on a continuous basis as the whole process and the whole landscape of bioequivalence develops in Africa.
So all the stakeholders should be at the table. Absolutely. During this harmonization. Yes.
Yes. Yes.
Exactly. Yes. So a question came in from Zada, and she or he asked, what is the current CRO landscape in Africa? What organizations are gaining ground in setting up BE studies infrastructure, and how can their experience be scaled up to other upcoming CRO centers in Africa?
So maybe I can jump in. The landscape, and I think I hope most people attended also the webinar that was put out day before yesterday by USP, indicating different business plans for CROs. And I think that work came about by realizing that the CRO landscape on the continent is very small compared to other places, for example, in India or even in the Middle East or in the West. And so considering the number of companies we have on the continent versus the CROs that are functioning and well functioning, and let's say maybe to a regulatory level, the landscape is very small compared to what those companies that need to do BE studies. So on the continent, the landscape is small. However, there are many efforts to increase the number of CROs on the continent, and that is part of some of the work that we've seen with USP. However, there are other neighboring countries, Egypt, North Africa, Egypt, Middle East, that also have CRO that are regulatory compliant, that can be able to perform the studies for us on the continent.
Exactly, exactly. So a few more questions. I'll also encourage the audience to send in questions in the chat box or in the question. Yes. So a few more questions are coming. Yes, yes. So we have this from RW. What are formulations where BE is not needed? What are the BCS, that is the pharmaceutics classification system classes? We do not need BE. Okay, so, and why are they not needed? And when is the biowaiver enough?
Yeah. Okay, so BCS biowaiver for class 1 and class 3, it is possible. It is not absolute, of course. It will depend on the drug product, on the dissolution results, on the linear pharmacokinetic. These are some very rough criteria, but if the drug is BCS class 1 and class 3, it is eligible for a biowaiver. Then there is a strength biowaiver, which means that if bioequivalence study is done, for example, for the highest strength and 3 strengths are developed, then the other 2 can be waived. Then there are products where it's not possible to do bioequivalence. These are locally applied products, for example, eye drops. Yes. If the eye drops if they're a solution, pharmaceutical data should be sufficient. If it's a suspension, then you cannot do bioequivalence study, but you do have to do a clinical study. Now it's changing a little bit. There is more emphasis on the quality data, but I think to a large extent the principles apply. Then, for example, we have topical products, ointments and creams. You cannot do bioequivalence on that. Then there are special pharmacodynamic studies which are outlined in the respective guidelines. So these are just some examples. Yeah, sorry.
Yeah, that's right. And as you asked the question for BCS waivers, there are guidelines, ICH guidelines that really spell out what products. For specific organizations, if you're interested in a specific product, you need to do your design, your formulation, and all of that. So you have to rely on some of the guidelines and also maybe a bit of training to ensure that you understand the formulation implications of your product characteristics for BE. And also BE does not always mean doing a study in humans. There are different levels of BE where you rely on pharmacokinetic data. I think you mentioned some PKPD, clinical endpoint studies, some products like BCS, you have the solution. So there's a hierarchy of what amount of data you need to generate for your product depending on its characteristics. So, combining both the responses will be able to give you a good idea of that.
Yes, it does. So we have a few more questions coming in. We have from Ndika. It says, can a BE study for a generic be going on while the exclusivity of a branded product is still valid? Or does it have to be when it's already expired?
Well, you can do a BE study anytime, but then the company, the originator, will go after you in the court. No. I mean, there is a Institute of Market and Data Exclusivity in Europe. It's 8 and 10 years. So during that time, nobody is allowed to, I mean, you can start developing a generic product. Nobody can prevent you from that, but you are not allowed to submit marketing authorization and you are not allowed to bring it on the market until the expiration dates. And
Okay.
actually, European regulatory authorities, they're very much aware of this, and they're very, very meticulous in checking the exclusivity.
Yeah. Okay. Dr. Lois, did you want to add to it, or should we go to the next question?
No, I think it's, as Marina said, the legislation in your country, in the jurisdiction. Do you recognize? I guess on the continent, you could say, does your country recognize the exclusivity that has been issued in another country or continent? And, that will bring other complexity. But in general, that is the rule.
Yeah.
Okay. Thank you so much. So this question is from Anesu, and I would like to admit that he is actually one of the scholars on the African Vaccine Academy program. So Anesu says, BE can be challenging in Africa and may lead to a lack of access for complex or sensitive molecules like cephalosporin acetyl. Are there any AI or computer models that are now acceptable in place of an actual BE study?
Well,
AI,
yeah. Go ahead, Lois.
No. I was just trying. So the question is: are there any AI models that can be
Used in
for
place of
BE
an
actual
actual
BE
BE
studies?
study? Yeah.
From a regulatory perspective, I haven't seen anything. I know we have IVIVC that can serve as a surrogate. However, I haven't seen any AI models. Again, as Marina said, it is the industry's parts to innovate and challenge the regulators. So bring it on.
Yeah. I think I've seen recently that EMA is now communicating some aspects of the use of AI in the regulatory world and the drug development world, but I don't think that we are that far yet that we can, with safety or confidently fully substitute clinical or pharmacokinetic studies with AI programs. I think we are still quite far from that. And I mean, the regulator being the most conservative, it will take also a tremendous amount of experience before we can surely say, no, we do not need bioequivalent studies anymore.
I do know the US FDA has some regulations around AI and machine learning. I also know there are some pilot studies that using AI to review regulatory documents if they're submitted in the standard format. However, nothing in place of the science of actually doing the studies itself.
Okay, thank you. So this is from Ahmed Yakasai, actually one of the key players in the pharmaceutical sector here in Nigeria. So he says, what are the common BE deficiencies? What are some of the common BE deficiencies that you could share here?
A lot of them come from there are various publications that have been put out related to common BE deficiencies. I know a lot of them lie within the bioanalytical data that's generated. A lot of it lie also on the quality of the dossier submitted, and then misinterpretation of data, dropping of studies when you study subjects when you shouldn't, wrong statistical. So it could be a whole day presentation on some of the common B deficiencies, but if you look at a lot of the studies, you'll see a high percentage of them are found within the bioanalytical space, and this leads to how you formulate, how you generate your data, which is then used to make the final BE decision.
Okay, thank you so much. Thank you. So this is from Dr. Chinedum Babalola. She's also one of the key pioneers in the pharmaceutical sector here in Nigeria. So she asks that, first, she thanks us for the webinar. Thank you so much. Then she says, since BE studies are invasive, are ways of promoting harmonization by the AMA/AMRH, etc. for Africans, We have a huge number of generics per product. Can BE streamline the numbers? Having carried out several BAB studies, PK studies, pharmacogenomic studies, how can one deal with variability in African projects?
Can you repeat the question again, just the last part of it?
Okay, so she says that we have a huge number of generics per product. So can BE streamline the numbers? Now, having carried out a lot of BE studies, PK studies, how can we deal with the variability in African projects?
So if I can reword the question is, we have a lot of products on the market for the same indication, and from product to product, we have variability within the product. So I think one of the key issues we may see with this product variability is maybe how the study was conducted. Maybe we have some substandard or counterfeit products in the mix masquerading as a generic version. And so one of the things, in addition to doing good and proper BE studies, one of the things is market surveillance and ensuring that the products continue to maintain their quality as they are on the market. The other thing is ensuring that companies, if there are any variations within their product that may cause variations in the BE, that these are handled within the regulatory pathway as it's outlined in the regulations for variations or changes after post-approval. So there are many ways to limit varying products on the market. However, the question really mostly addresses post after they've been approved. However, products coming on the market should meet that requirement. And some of this could also be linked to what reference you've selected and how that reference varies with company X's reference and why they selected it. So a lot of it has to do with properly designing your study, properly having the right reference. And once a product has been approved on the market, making sure you have surveillance to ensure that the product is not changing over time, especially with the quality, ingredients, GMP, sticking to GMP, and all of those other requirements. I hope I answered the question that was being asked.
Yes, yes. Thank you. More questions are coming in, and we have this from Janet. What strategies are you considering to implement to remove the BABE related roadblocks as per the webinar is based on? So what strategies are we considering to remove these roadblocks?
I think, go ahead, Marina.
No. I mean, there are many roadblocks, and there will always be roadblocks, right? And as the environment is developing, then we all start realizing that there are different hurdles. No system is perfect, but what is important is to maintain the general idea and concept of what we are trying to achieve here, and then go one step at a time. And as we were mentioning before, in relation to the harmonization, what is important is the intense dialogue between academia, between industry, and between the regulatory authorities. What is important is to work on capacity building and to motivate regulatory authorities to achieve ML3, because this will bring more transparent and more predictable environment for the industry to, and it will be more attractive environment for the industry to start investing in the BE areas. And of course it's a long-term process and it takes time, but yeah. So Lois, you were going to say something as well.
No, I think you said some of the things I was going to say in terms of capacity building, getting support from the regulator in advance of maybe designing your studies, getting your protocol reviewed, getting training if you're within the CRO space to ensure that you're conducting adequate studies. So I think you've covered it sufficiently, Marina.
Thank you. So we have this question from Abubakar. Can we also use bioinformatics information, take for example, DNA or protein data to run BE study? Can we also do that?
Well, wow. It depends. It depends how. I mean, the regulation in terms of BE studies doesn't cover the background and the search that each CRO needs to do in order to design the bioequivalence study. So I mean, if the and many CROs actually when they're approached by a client with a proposal for a new bioequivalent study for a molecule, the first thing that they do is that they learn everything that they can about this molecule, and they dig into the literature and try to characterize the active substance and so on and so forth. And I mean, what sources do you use for a design of a bioequivalent study? It's only your business. No regulator can say this is wrong. It's not their job to do that. But it is also the risk of a CRO, of course.
Yes, exactly. If DNA or protein data is necessary for you to show BE for your product, and it's the only way you can show BE, then you can submit that data to the regulatory authority. By the legislation and regulation, they're required to review it and ensure that how the data was generated is reliable, validated. And if you can show that you can prove BE in this form, then yes, of course you can do it. In my experience, I haven't seen it yet. However, innovation keeps advancing every day.
Yes, thank you so much. One more question from Kevin. It says, how can we make COA more available to suppliers to help patients access to essential medicines here in Africa?
Is that COA certificate of analysis?
Yes.
I think the COA is generated based on the information that's submitted by the company, how they've tested their product, what critical parameters are being tested. So we're making it more available to suppliers of medicines. I think this is a conversation between the supplier and the pharmaceutical company and how they can make it. There's no regulatory requirement to force a company to submit the COA. It's good information to have. But yeah, I think that's all I can say about that.
Yes. So we have just two more questions. I know we are running out of time really fast, but they are actually quite interesting questions. So we have this question from Oluwole. It says, do I need BE study for newly formulated oral solution generic product that is not in BCS class 1 or 3 and that has no comparator product?
Oh, well, that's
That's a side conversation.
Yeah. Okay, when there is no comparator, you cannot do the bioequivalent study. That's clear, but it doesn't mean that you don't need to do anything. So, for example, European practice is if you have a solution, there are a couple of regulatory pathways that you can choose. The worst possible one that no sponsor wants to hear is to perform a clinical efficacy study in the target patient population. There are other pathways as well. That it's Article 10A that we have in Europe. It's called it's the well-established use, and no data need to be generated, but there are strict conditions. that needs to be fulfilled if the product is to be qualified for a well-established use. Then it depends if the solution is intended for systemic action or only locally acting somewhere in the gastrointestinal tract. So yeah, I think it's a consultation on its own, very specific one. But there are a couple of ways to how to go about it.
Yes.
I think what Marina you summarized is similar to what happens also in the US, and also could be a reliance mechanism for companies conducting BE studies on the continent. Bioequivalence is a comparison study. So if you don't have something to compare it with, then you can't do a BE. There's a hybrid situation if you have access to that clinical data. And as Marina said, or you have to do the clinical study, a clinical endpoint study, a bridging study to show that your product actually works in that indication.
Yeah.
Yes, exactly. Exactly. So we've come to the end of the webinar. It has been a quite intensive and insightful, a little bit over an hour now. Thank you so much to our experts, Dr. Lois Kikwai and Dr. Marina Fetik. Thank you so much for your insights, and also to the audience who have stayed with us this long and sending in such insightful questions. Thank you. As we've explored, mandatory BAB studies are a fundamental pillar, and it's good for Africa, especially with our ML3 level, to align as quickly as possible. And we have the experts here that can handhold us throughout the entire process. So the Bioequivalence Training Initiative, which is what has brought this webinar to us here today, stands ready as your partner in this vital journey. Our team is deeply committed, as you've seen, very informed, very knowledgeable about the space, not just in Africa, but also in Europe. And we are deeply committed to providing the expertise to successfully navigate this evolving landscape. We are specifically collaborating with regulatory agencies, pharmaceutical industries, professionals, to effectively remove these roadblocks and ensure that we have a clear path to quality medicines in Africa. I have left my email and pinned it in the chat box, so please send me your questions and also reach out to us if you want to take this a step further, and we invite you also to engage with us and see how we can help you navigate this landscape as it evolves. Thank you so much to everyone. Our speakers, our experts, you want to say just a few words to the audience before we call it a day? Over to you, Dr. Lois, Dr. Marina.
I just want to thank everyone for attending the session. We had a very big crowd. Thank you for those of you who asked questions. There are many questions, and I think we can work together to alleviate some of the questions. And as Toon said, send your questions to her, and we'll be able to respond back to you in a timely manner with some responses and answers to your questions. We're also available for any further consultation that's needed.
Yeah, I think Lois summarized it very nicely. So the only thing that I can say is thank to everyone. It was a very nice discussion, and I hope it was helpful, it was insightful, and that it's not the last one.
Yes, thank you so much. Thank you to everyone that joined us, and we'll definitely send you the slides and the link to the recording as soon as it's ready. Thank you so much. Have a wonderful rest of the day, everyone. Bye.
Bye.
Thank you. Bye.
Thank you. Bye.
Thank you. Thank you.
Thank you.
Thank you.
Bye.
Thank you. Bye.
Bye.
Thank you. Bye.
Bye. Thank you.
Bye.
Thank you. Bye.
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 Thank you. Bye.
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 And we are talking about it here.
Very well.